So thank you, Francois and Julie and all of the teams. It's been so far an amazing visit. I've really enjoyed it. It's one of these people who you have known their work and who've been working in the similar projects and similar areas for many decades. And it's to me, it's shocking that it took until last year that we could sit down and have a beer and a glass of wine and then another glass of wine. And then we were at the EASD in the members lounge, and we hatched the idea. I said I come to Europe a lot, and so we hatched the idea of this visit, and it's been an amazing pleasure and such a dynamic group, so many interesting projects, and I've really enjoyed it. And of course, the food and the hospitality has been fantastic. And I apologize that this lecture is in English. As I said, my restaurant French is actually reasonably good, but my science French is not good at all. So for the sake of communication, I'll be delivering in English, but uh, I'm very happy to be here. So this is Vancouver. My condo is actually by the bridge there. So I'm very fortunate to live in Vancouver and the mountains. People assume that Vancouver, because it's in Canada, is cold, but actually it's warmer there than it is here, certainly now. But this cold doesn't bother me. The title here is a short version, but it's the same topic that I would talk about. I also have a role as the Vancouver Coastal Health Research Institute Associate Director, and so it was cool to see some of the facilities and think about these ways to collaborate as institutes. Okay, so I don't have any conflict of interest, but I do like to tell people that I once did work for Novo Nordisk, and this was now a long time ago. I ran their institute in Oxford. And then also I helped start what is a charity now in Canada called the Institute for Personalized Therapeutic Nutrition. And that's leveraging some of our work and our interest in hyperinsulinemia and how different nutrients regulate insulin secretion. But it's a volunteer position, and I'm certainly not going to get rich out of charity. I also always like to acknowledge that in Canada, being a colony, that I work and I live on the traditional, ancestral, and unceded territory of the Musqueam people. Of course, when you grow up, you don't learn too much about how the country came to be. It's a wonderful country, but we need to think about these things. And it's also very important in diabetes because the Indigenous people in Canada and the US have a very disproportionate burden of diabetes and related metabolism diseases. So the work we do hopefully will benefit them as well. Okay, so today I will basically break my talk into two pieces. One is we will talk really briefly about our work on hyperinsulinemia, because I know it is of interest to the group here. But most of this is published, so I won't spend too much time on it. I'm always happy to send the papers. We have time, of course. I'm happy to answer questions after the talk. And as you all know, I love to talk science. So anytime you can email me, you can send me a message on any of these social media sites as well. But we'll talk about the consequences of hyperinsulinemia, why it's important to understand insulin secretion. In this talk, I'll talk about obesity, I'll talk about fatty liver, I'll talk about insulin resistance. Not in this talk, but we've done this work. We've demonstrated a causal role for hyperinsulinemia in longevity, in pancreatic cancer initiation. Also, we're working on breast cancer initiation. We dabble in the heart and hypertension as well. But the last part of the talk, which is more recent and includes some unpublished work, we are going to talk about how insulin secretion is regulated, obviously at baseline and stimulate insulin secretion, but not just by glucose, by amino acids, by fatty acids, ketone bodies. What I won't talk about today, but we have wonderful collaborations, is that insulin secretion is also regulated by pollutants in the environment. It's regulated by the number of beta cells and how healthy and happy they are. I won't talk too much about our work in beta cell apoptosis and beta cell fate and beta cell states, but you can find our papers online in that regard. So the concept, of course, starts with how important type 2 diabetes is. Every time we add these numbers, they become obsolete because there are hundreds of millions of people around the world who live with type 2 diabetes. There are probably twice as many people, at least, that live with prediabetes, and then there are twice as many people with that who live with overweight or obesity. And let me see if this mouse, it shows, okay, good. So, when we talk about diabetes, we're all the way over here, and there's not enough insulin to meet the glycemic demands. Insulin resistance is high, especially in type 2 diabetes, and you often have adiposity. But, this is kind of the end stage. Um, we we are beginning to think about how we can modify the course of the disease in this prediabetes space. Here, you have not yet fully had dysglycemia, but you have insulin resistance, you have hyperinsulinemia, and adiposity. Where I think it's really important in the prevention and to really make an impact is understand what's happening here. And in those early stages, a lot of things are changing. You have hyperinsulinemia, you have insulin resistance. Of course, many people think that the hyperinsulinemia is caused by the insulin resistance, and that might be true, but I'll present some evidence that the other way around is also true, where the hyperinsulinemia can cause the insulin resistance and cause the obesity. And at this state, by and large, glucose is pretty normal. So we can remove glucose from the equation. But this is the sort of the common traditional model where the insulin resistance and the obesity are ahead of before the hyperinsulinemia, and they cause it, and it's a reaction. Okay. But if you look at these old studies as well, and these are some of this is a picture taken from one of the earliest studies comparing impaired glucose tolerance and normal glucose tolerance. And here I've just cited a review that we put these in. The defect in insulin secretion, even in impaired glucose tolerance, is actually quite interesting. So of course, there is a hyperinsulinemia. There's a delay in the first phase. Sometimes the second phase is delayed as well, and then it stays hyperinsulinemic here as well. And that results in this mild impairment of glucose. So the first question I will hopefully address for our group today is, can we demonstrate that hyperinsulinemia is causal in obesity and insulin resistance? And I always like to remind people that none of these ideas are new. I've helped popularize them. I've added some genetic evidence, but we really stand on the shoulders of giants. And one of my favorite people and favorite colleagues is Barbara Corky, who celebrated her birthday the other day. I sent her a message on Facebook. But Barbara was so far ahead of her time, and back in her Banting lecture in 2011, she asked the question, hyperinsulinemia, cause or consequence? And in her mind, it's a cause. And then I also like to mention this article here, and this is Walter Pori's, a pioneer of bariatric surgery, and I'm not an expert in this area, but he also asked this question, is it hyperinsulinemia that's the culprit? And the reason he asked this question is when they did the bariatric surgery, one of the first things that was corrected is the hyperinsulinemia before anything else, before the weight change, before the insulin resistance, before the glycemic improvements. So that seemed to be good evidence. So what do you need to show that something is causal? I'm going to give you the human data, that correlative evidence first, and then I'll show you our mouse data. So one thing is you would want to see that the hyperinsulinemia happens before or even in the absence of insulin resistance or some of these other factors. And that evidence, some of it comes from a really nice bit of work from a group in Italy, and this is Domenico Trico, who many of you know is a sort of a rising star in the space. And they identified a group of adolescents who have hyperinsulinemia, primary hyperinsulinemia, they called it, which is present in the absence of clamp-measured insulin resistance. So it can be prior to this. And then the follow-up paper from this is actually quite nice, is that those individuals with primary hyperinsulinemia, they had more adiposity, larger adipocytes, and we wrote a commentary on this in diabetes. So it can be correlative predictive. There's other studies that suggest this as well. And then there's some other studies that in other cohorts don't find the same correlation, and that's fine. As we discussed yesterday, it's quite possible that there are different, well, we know there are different subtypes of type 2 diabetes, different subtypes of obesity, and now we know there are different subtypes of metabolic liver disease. So, so what we're saying is this might not be for everyone, but it certainly is for some people. And then the other question is a very obvious question, is an increase in insulin by itself with no other variables, all other variables the same, is this sufficient to cause weight gain and insulin resistance? And the answer is, of course it is. We have these huge cardiovascular outcome trials with basal insulin and long-lasting insulin, and every single one of them, there's an element of weight gain. And so that's everything else is controlled. There's thousands of people. And so certainly it's necessary and it's sufficient. So what about the necessary? So people always ask me, is there any evidence in humans that blocking hyperinsulinemia can cause weight loss or improve insulin sensitivity? And there is. It's a bit difficult to do. There are not that many drugs that specifically inhibit insulin secretion. You can argue none of them are truly specific, but diazoxide, if given at the right dose, can have beneficial effects in obese hyperinsulinemic adults. There's a few studies on this, and same thing with mice. And there's also studies using triatride, somatostatin, to reduce insulin secretion. But these aren't really super specific. And similarly, there's various diets that can lower insulin. They can have effects, but they're not specific. They don't tell you, it's not insulin that's the only thing that's being reduced. And to do this, of course, since we are not about to do genetic experiments in people, to do this, we need to use mice. But I'm very confident that the mouse research is actually very relevant, very applicable because of evolution. Insulin is an extremely well-conserved hormone. It does the same thing in worms, flies, mice, humans, dogs, pigs, monkeys, you name it. And millions and millions of years of evolution, it is basically a hormone. It's very conserved, does the same thing. So how did we do this? And I'll show you a few different configurations, and it's important to understand the logic of this before we go forward. So mice and rats are unique in the animal kingdom in that they have two insulin genes because of a duplication of the insulin gene that has happened since separation of, for example, guinea pigs only have one, and we only have one. The insulin gene that we have is homologous to the mouse or rat insulin 2 gene. But the first study I'll show you is with this duplicated insulin gene, the insulin 1. And the reason we're doing this here is that this is a simpler gene and it is only expressed in the pancreas primarily. So in this first study, we have gotten rid of the insulin 2 gene, which is in the brain, the thymus, and the pancreas. And we are studying mice that either have 2 alleles or 1 allele of the insulin 1 gene. And on a chow control diet, you can see here in blue that even the mice here that at 1 year, if they have the reduced insulin gene dosage, they have about half as much fasting insulin. And here you can see that these mice that are fed a high-fat diet, they become very hyperinsulinemic. But if they don't have the extra insulin allele, if they have a single insulin allele of insulin 1, they are genetically incapable of sustained hyperinsulinemia. So here at year 1, these mice, it's as if they have not even seen the diet. They have sustained low insulin. So this allows us for the first time really to separate the effects of insulin from all of the other effects. And this is the bottom line, the punchline. Mice that are genetically incapable of hyperinsulinemia are incapable of gaining weight on the high-fat diet. So this means that the hyperinsulinemia is the cause of the weight gain. And this is really interesting because in this first year, this all occurs in the absence of any really major changes in glycemia. There is a period here where the mice with reduced insulin, and these are sort of young, very rapidly growing mice at 11 weeks, they have some glucose intolerance. And actually, if you go down to that single allele of INS1, and especially in some of our more modern facilities, some of them will become insulin insufficient. So we're right on the edge here. But you can see here that the glucose is generally normal, especially later in life. The response is pretty normal. Glucose response is pretty normal. And the insulin sensitivity is pretty normal Actually, if you look carefully, you can start to see that near the end here, they're actually slightly more sensitive because they have less insulin. So here I will define operationally, we are going to define hyperinsulinemia as insulin levels that are in excess of what are required to sustain glucose homeostasis. That's my operational definition. And there's a lot of stuff in the literature these days, especially with the GLP-1 drugs. People think, oh, the only thing that is important in glucose homeostasis is weight loss and things like that. I think that is an oversimplification. It might be clinically useful, but mechanistically, although weight and glucose homeostasis are co-regulated by many factors, they are mechanistically separated, and we can easily do this here, right? Okay. So the mechanism of this, at least in these mice, which were held at room temperature, which we know is not the most sophisticated way now of doing it, and we're redoing some of these things in thermoneutrality, but this was largely due to an increase in energy expenditure. And importantly, the energy expenditure was prior to the weight differences, and it was in the absence of any major effects in food intake. We later do some pair feeding studies. We can see some small food intake effects, but it's effect. And we were surprised by this because this was a long time ago, it was over a decade ago now, but we sort of accidentally stumbled across this idea of browning of the white adipose tissue. And this was actually an accident because we had these arrays of qPCR, and we were cheap because we're a Canadian lab. And so we bought the same array to use in all the tissues. And it was a surprise to us that we saw UCP1 and many of these other factors, which we were intending to look at in the brown adipose tissue, in the white adipose tissue. But certainly we can see it at mRNA level, at the protein level here as well. We have a little bit of an echo, but it's from the Zoom. No, I think it's because the Zoom had a delay and now it caught up. I think we're okay. It also makes me sound a little bit like Darth Vader, which is all right. And then in more recent papers, we've studied these again. We can see an upregulation of mitochondrial enzymes. This I included here. In these mice with reduced insulin gene dosage, they're completely protected from the fatty liver and circulating fatty acids, cholesterol, things like that. And they're also protected from the inflammation. And whether this is direct or indirect, it depends on how you think, but we can see this in many tissues. So the conclusion for this first part is hyperinsulinemia is required for diet-induced obesity, inflammation, and for lipid spillover. And instead of being just a consequence of obesity and insulin resistance, we show in this model that it is a cause of these things. We don't address the insulin resistance in this model, but we'll do it in a minute. And this tells us that we could potentially, if we intervene early enough, we could have therapeutic benefit by reducing hyperinsulinemia, which sounds counterintuitive, of course, because insulin is so important for glucose control. You obviously never want to have not enough, but I think many of us live with too much. So this is kind of a cool thing, but this is a reduction in insulin for the entire life of the mouse. We also did a fun trick, and this was a postdoc who was in the lab for a little while, who now works in Belgium, in the French university outside of Brussels. And actually, she left there. She didn't know how to speak a word of French, so she's had to push on through. So this was a cool experiment. Here we take mice, we get them fat first, and then we use inducible Crelox to reduce the insulin gene in an already obese mouse. And this we can do without affecting glucose homeostasis. It has a minor effect on insulin secretion and insulin mRNA, and it reduces the size of the mouse and the fat pads quite quickly. We believe this is relevant because of the conservation and evolution. But actually, my colleague, Liz Rideout, did the same thing in fruit flies, and you can see the same effect on the fat pad of the fruit fly. So I think this is probably relevant. So I didn't show you the data, but the mechanism here is that the insulin seems to regulate a PTRF, which is also known as cavin, which is actually a human lipodystrophy gene. So we know it's relevant in human adiposity. So what about insulin sensitivity? We show a clear correlation in human samples. This is it's a negative correlation of insulin levels and the insulin receptor mRNA. So we know that high levels of insulin downregulate the insulin receptor. This is published in Howard Sen's FASEP paper. And then there's a beautiful study by Justin Gregory, and he was actually able to show that insulin treatment in people with type 1 diabetes causes insulin resistance. And for a long time, this was controversial. And they said, well, maybe it's the glucose. But he used very cleverly, he used controls of glucokinase moding that have the same glucose. And the only difference between these is that the insulin is either added or higher or lower. And in fact, it shows that. So let's go to our mice. Now, this is the different configuration. It's actually a slightly smaller change in insulin. These mice have no insulin 1, and they have 2 alleles or 1 allele of insulin 2. And this was part of a longevity study. So we followed these mice their entire life, 2.5, 3 years old. At 2 years, you can see the fasting insulin is reduced. It's not half, but it's maybe a third or a quarter. And the glucose homeostasis, these are the different diets. The two genotypes are perfectly on top of each other, except at the very beginning, where mice with less insulin have slightly higher glucose. After that, they adapt and they are exactly the same. The diets are different, the mice are the same. But if you wait long enough, the mice with less insulin have lower fasting glucose. And the reason is that they are more insulin sensitive. And this makes sense in sort of general endocrinology. The more you have of a hormone, the more you downregulate the receptors. It's very easy to do. Diabetes is the only field where we think that the resistance to the hormone comes first and that the ligand somehow compensates. I'm just saying. And I was originally trained in the sort of pituitary endocrinology. And this is beautiful work done by Nicole Templeman when she was a graduate student in the group, and then she went and worked in Worms, C. elegans, and Princeton, and now she's a professor in University of Victoria. And we also can see this much faster if we add sucrose. We can see that, that it's also critical for insulin sensitivity. So this tells us that the insulin hypersecretion can be the cause of insulin resistance, in conclusion number 3. So let's switch gears a little bit here and think about what are the interventions that can reduce hyperinsulinemia. And so this, I love this study. This is a paper in Nature Medicine, and it is, I have to put my glasses on here because it's a really huge number. They studied 205,000 healthcare professionals from 3 US cohorts over 32 years. And so what they did is they looked at all these different types of diets that people were given. And this is actually an interesting thing. And it turns out the diet that was the best for just about everything was the diet that was designed to reduce hyperinsulinemia. And there's actually quite a depressing thing in this slide too. This diet here was designed by the World Cancer Research Foundation and the American Institute for Cancer Research to reduce cancer. And it did not do very well. So a lot of what we thought in nutrition, it turns out to probably be wrong. And the diet that was the best for metabolism and for cancer is the one that reduces hyperinsulinemia. The other thing, of course, is there are lots of things, lots of interventions that can reduce insulin: bariatric surgery, very low carbohydrate diet, very low calorie diet, intermittent fasting, ketogenic, blah, 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 all these things. And many of these things reduce the insulin prior to the weight loss. And I was fortunate enough to be part of this very interesting trial as sort of an advisor. And these are the people also in the IPTN that helped push this along. It's a cool trial in part because it was delivered by pharmacists, which in the rural communities of Canada have more time with the patients than the physicians do. And it is a low carbohydrate, also low calorie, ketogenic diet, 200 people. This was done with very little money. So the big problem here is it's actually quite short. But amazingly, you can see that 36% of these people, and they were on, they took all comers with established diabetes, some of them on 10 medications, 30%, 35% were able to come off of all of their diabetes medications. very, very quickly and prior to the reduction of weight. So this tells us that if type 2 diabetes is reversible and remission is possible. It's been shown, of course, with surgery, but we do know it's possible with diet. And so reducing carbohydrates is one way we could reduce. Okay, so this last part of my talk here is really focused on, will this work for everyone? Okay, so you guys are experts in human islet, and we were just talking this morning about how much variability there is in the responses in vitro and, of course, in vivo. And this is shown in vivo. This is work by Sarah Berry in London. And it's just an example of some glucose and some insulin traces in just a lot of people. And it's just striking. In contrast to mice, which behave, humans are all over the place. See, whatever they ate, whatever's in their genetics. This is 1,000 patients, and it's just a super variable. So the answer is not everybody responds even to glucose the exact same way. And even an individual person on 3 different days will respond differently to the same glucose load. Okay? So this is work done by an incredibly talented postdoc who's just obtained a faculty position. She had lots of offers, actually. Yelena Kolic. And what she did for about 7 years, except one maternity leave, someone else helped her with it, is we took every batch of human islets that came from the Pat McDonald's group in Edmonton, which is actually where she did her PhD as a patch clamper. And we did a standardized analysis of the response to glucose, to a model amino acid, leucine, and to fatty acid model oleate palmitate mix. And from each of these, we also saved an aliquot, which we did deep mass spec proteomics on. And then there were aliquots sent all over the world, and we have access to that data. There was RNA-seq done by Anna Gloin's group. There's a nanostring done by Francis Lin, lots of things. And this is the distribution of the donors in this study. One limitation is it tends to be male skewed. So analysis of male versus female is not so good. This tells you a little bit about how reckless maybe some of the male drivers are in Canada and whatnot. But this is one limitation. We can't make very strong sex differences with our data. Okay, so let's start with the averages. And this is maybe 140 donors on the one side normal and maybe 22 or so of type 2 diabetes. We have now another 100 that are in the freezer that we will add to this. Of course, this is all online, as you know, in the humanislets.com website. But I think even adding another 100 is not going to change the shape of these curves. This is now, for us, in our system, kind of a ground truth. So the brown thing here is the response to glucose in normal. You can see nice first phase, nice second phase. We also gave 6 millimolar glucose, which is much more physiological. And then we gave KCl here. And in the orange, you can see the response to leucine. And then you can see the response to leucine. We weren't sure if these things would be effective at 3 millimolar glucose, but we did it on top of 6 millimolar just to see the sort of synergy if there was. And you can see a little bit of synergy here, although it gets to the same amount. And then this is the mix in blue of oleate palmitate. And on average, again, this is a small effect that you can see. And I was actually most of your parafusions are also 3 to 15. So theoretically, I mean, your flow rate is a little faster, but theoretically, we could compare notes. In type 2 diabetes, we see the textbook traditionally well-known loss of first phase, and the second phase is a little bit lower too. And then actually statistically, the response to fats is lower. But the response to amino acid is not statistically lower, which is of relevance because in my meetings I just had in London, working with Nicola Guess, we're interested in potentially using this amino acid response, which is preserved in type 2 diabetes, leveraging it. KCl response is a little bit lower as well. We can compare the transcriptome and the proteome from type 2 and control islets. And then we can compare, it's from the same donors where it overlaps. We can look for where there are differences in both. I will tell you right now that in our hands, the proteome is more stable and provides more robust and bigger differences. But we don't see everything. So we see 8,000 proteins. There's probably 4,000 that are maybe in the membrane. They're hard to see and stuff like that. So it's not perfect, but it's really powerful. Somewhat disconcertingly, the correlation on a per gene product basis of RNA and protein is only about 0.5. Maybe that's good, maybe that's bad, but you can't assume it's protein when you're looking at RNA. And this is just a way of mapping the differences in type 2 diabetes, the protein-protein interaction networks. And you can see some really robust differences in the mitochondria, wherever that is, in the ER and ER to Golgi trafficking, some very famous things, some unknown things. We were talking about this, it would take me and my, 25, 30 people in the lab, 100 careers to follow up on all these proteins. So, this is why we put this online. Everyone can find their protein. They're all very significant. But what I really want to talk about today is how we've leveraged the variation. Those were the averages. And the averages here, those exact same traces are shown down here in the blue and the salmon. But the distribution of the individual responses is incredible, right? And we don't see, look, some of the fatty acid responses are nothing, and some of them are huge. And as you'll see, there were some people's islets who had a higher response to fatty acids than to glucose. We just cannot model this in the mouse. We do a lot of stuff in mice. We think mice are incredible models, but their genetics is so similar to each other. We can't get the variation. The diet is identical. These traces are from mice of all different ages, male and female, just pooled together. We can't model this variation except in human islets. So we are taking advantage of the variation. And it's in the paper, but we can show the correlation of every protein with responsiveness across the individuals. And as I said, there are protein hyperresponders or amino acid hyperresponders. We know that there's only one or two. There's two proteins that are associated with low response to amino acid. But what we did here, or what Yelena did, is she separated the responses that were higher than that person's glucose response versus all of the other ones. And we're following this up in a separate unpublished work to try to understand every amino acid. We just did leucine, but nobody's really done this in human islets to really compare the 20 main amino acids. And this is in control, and it's only a very pilot study in type 2, but you can see the results here. We were really excited about this because, again, most people don't think human islets or any islets respond to fats. And if you take the average, that's true. But some individuals are hyper responsive to fat. And there were quite a few very interesting protein differences in these things, including, you'll notice, Wnt4, AKT, on this side, HNF1 beta, that are associated with high and low responders. The reason this is translationally interesting, of course, is that not everyone responds to these, for example, ketogenic diets that are designed to reduce insulin. If you told a person, hey, you should go have this high-fat, high-protein diet to reduce your insulin, but they were one of these sort of 8 or 9% of fat hyper-responders, their insulin would go up instead of down. And then they would complain on the internet and they say, oh, this diet sucks. This doctor is not very good. This nutrition is not very good. But it's their physiology that's different. We also have this interesting thing. We work a lot with the stem cells. It turns out that the stem cells respond to the fatty acids before they respond to the glucose. It's actually quite interesting. And it is just a minor story that we published, but we're interested in other non-nutrient or pseudo-nutrient stimulation. This is our ketone. So all of this work was done by Yelena and her team of undergrads and friends. So this is interesting. This is the response to ketones. And it's interesting that the response is dependent on BMI to quite a high degree. So the conclusion from here is that there's a lot of heterogeneity in the response, and the proteome varies with disease and with the nutrient responsiveness. And so eventually we would like to translate this to clinical practice, and we're working with the IPTN to do practice change, working with government, working with training of physicians and things like that. So the conclusions overall is that lowering insulin can reduce adiposity, liver fat, inflammation. Hyperinsulinemia can be a cause of insulin resistance by downregulating the insulin receptor directly, probably by many other mechanisms. Mice with modestly reduced insulin production, they live longer. We showed this as well. I didn't show you the data today, but it's very powerful signal for pancreatic cancer initiation. This is Janelle Kopp and Annie Zhang's work. And then the nutrient-induced insulin secretion is highly individualized. Of course, these are islets outside of a person. So we are trying to find some funding for a clinical trial to do this in people. Believe it or not, it's actually not known in healthy people, side-by-side comparison of glucose, amino acid, fatty acid, how this would turn out. So we're interested in this. We're also doing some genetic analysis because we have the genetics of these islets to see if we can identify a gene that is associated with this response. And so, of course, we now have this resource. Everyone is so welcome to use humanislets.com. And then we're also very interested to hear from you, the users and the colleagues, what you would like to see in version 2 of humanislets.com. If you're clicking on it and you go, oh, I really wish I could find this easier, tell us, and we will try to add it in version number 2. So nothing I said today is to say that insulin is good or bad. Insulin is just powerful. If you don't have enough insulin, of course, you have diabetes. But if you have too much insulin, you can have all of these other problems. So with that, I want to thank all of the people, of course, Elena, all the people in my lab, all the people who've been in my lab before, my wonderful collaborators, the funding agencies. The picture behind is a picture of my traveling hockey team. And tomorrow I start my 9th tour with this group of folks I met at Oxford. This is us winning a trophy in Vienna. It's called the Oxford Canadians, and it commemorates one of the first Team Canada that used to travel to do the World Championships in Europe. And France was actually one of the inaugural members of the International Ice Hockey Federation. So they would come and they would play France. And it wasn't people traveling from Canada. It was the students, whoever was in Oxford at the time on a Rhodes Scholarship, who would form a team and come and play in Europe. And so we're proud to carry on this tradition. I like to show this slide here because it tells you how much I value teamwork and, of course, winning. I'm the goalie in this picture. But with that, I would be absolutely delighted to take any questions. Thank you for your time and your attention.
Thank you so much, Jeff. It was really wonderful travel around your work for many, many years and decades, in fact. So I'm sure there will be some questions, and I would just not wait until questions coming.
Okay.
Okay, just please. We have like 15 minutes of class, so please just be very,
Go ahead.
very free.
Thank you very much. I was wondering if the effect of the protein on insulin secretion is a direct effect, or if it eventually caused by other mediator, like so I have in my mind that proteins and arginine, for example, stimulate directly GH hormone, growth hormone. We can stimulate the cells. So
yeah.
The heterology could be.
So in these experiments, so the question was, is the protein effects direct? And so I want to repeat it for the Zoom people, well, which is just us. But so of course, these are isolated islets. So in this case, it has to be direct. It does not mean that there are not also many indirect things. So these are direct effects. We are not sure if it's just the beta cell or it's the beta cell and the alpha cell and them together. I think there is more we need to understand. So at this time, we can just say it's the islet. But we do believe these are direct. And you may have noticed, actually, one of the most significantly different genes in type 2 diabetes was a protein transporter, ARG2. And so we have, a lot of strong evidence for that. So I think the direct action of protein in the islet, probably also the beta cell, is a very, very strong and the alpha cell as well, is a very strong mediator of insulin secretion. And of course, we don't eat just glucose and leucine and oleate and palmitate. I hope not. So these things normally in our life come together. But the mix, I think, is very important. Yep. That's a great question. So the question is about pre-culture and about our culture conditions. So our islets travel to us in about 10 millimolar. They live overnight in 10 millimolar to be happy. And then they go on the perfusion rig, and we wash them with 3 millimolar for about an hour. So they're kind of fasted, if you will. But everything I showed you there is acute. So these are the response of the amino acids, the fatty acids in that minute. We have other studies where we look at the effects of them overnight. Of course, if you give oleate palmitate at higher doses for longer periods of time, you can have lipotoxicity, ER stress, apoptosis, and things like that. But everything I showed you here is the direct acute effect. And I also want to stress that the proteomics is not the effects of these treatments on the proteome. It is the correlation of the response to those treatments with the proteome, right? So everything is acute. At the back.
Yeah, thank you for this really nice presentation. Just 2 questions about insulin and insulin. Is that different in the regulation and the hyperdialysis? Are they both expressed at the same level or not? And the second one is in your model of INS1 deficiency. Did you look at the beta cell identity and or the differentiation and the effect of that gene dosage on the identity?
These are great questions. So the first question is about the INS1 versus INS2. How are they different? How are they regulated? And so the answer is, so first of all, the sequence is nearly identical. It's different by 2 amino acids. We think that they are similar potency on the receptor. We think they're pretty similar. There might be some subtle differences, but I worked with Novo Nordisk early on. You can get from them, they've synthesized INS1 and INS2, so we can compare. Their regulation is similar. Insulin 2 is a much more complex gene. It's the natural gene. Insulin 1 was made by a retrotransposon that flipped over into another place with just a bit of promoter. So the regulation of insulin 1 is in the same direction always, but it's less complex. Insulin 2 is the same that we have. It's the ancestral gene. It has very complex regulation. But sometimes you can find things that regulate insulin 2, but not insulin 1, and vice versa. It's a little bit complicated. If I have to trust one, I always trust the INS2. It's more similar to humans. The question is that you ask, can you de-differentiate a cell just by removing insulin? So we actually have a cell metabolism paper where we did exactly this. We took beta cells in vivo that had no insulin 1 and the floxed insulin 2, and we deleted all the insulin. Actually, of course, the mice will eventually get diabetes and die, but they did not really de-differentiate. So the insulin is like the last thing in the differentiation. It's not a major regulator. What happened, which was really interesting, is that these beta cells had less ER stress than normal. And what that means is that in a normal beta cell, just the production of insulin itself is itself so stressful that it's a hard to fold protein. The beta cell makes more insulin mRNA than every other gene combined. 60% of the mRNA in a beta cell is insulin. Everything else is actin and whatever is 40% combined. This is very stressful. What was cool is, and this tells us this is related to type 2 diabetes and type 1 diabetes, when we reduced the insulin, it released that ER stress. And actually, the ER stress is a brake on proliferation. The cells started to divide. But they never changed their differentiation status. Not really. Good question, though. Yes. Isabel.
Because it's very weird. Insulin is a trophic factor.
Mm-hmm.
In general, my understanding from the downstream signaling within the beta cell, from the insulin receptors, that it induces proliferation.
It does?
So
this is good. Yeah.
You also induce proliferation.
Yes. So the cell autonomous effects of insulin within the cell are, like I said, it's stressful. Reducing it reduces ER stress, increases proliferation. The autocrine effects of insulin, as you're suggesting, are actually the opposite. So we actually also studied that. And in my postdoc, and we worked on this, and it's actually a bell-shaped curve. At low doses, it's stimulatory proliferation. At high doses, it's not. And at same thing with apoptosis, same thing with regulation of various different RAF, ERK signaling pathways versus AKT. It's all dose dependent. And another very talented postdoc in the group, Sus Skovsa, when she was in the group, basically redid that classical experiment of the insulin receptor knockout in the beta cell. And we can see really interesting effects. So there's the autocrine effects, and then there's the cell autonomous effects. And then in real life, they are always playing with each other. Great question, though. And then, yes, keep going.
Maybe just about the human islet study, did you look at so glucagon secretion, so metastatin, you know that all those guys are also playing over secretion. Did you, and also the general heterogeneity of the islets, is there more alpha or delta cells within the differences in the individual?
Yeah. So two questions. One is, did we look at glucagon, somatostatin in the perfusions? We have not yet. We have all of the samples. I have a student who that will be their project. The experiments weren't designed to look at glucagon. Really, we don't go down to one. We don't have, but we think we can pick, in some studies, we pick just the peaks and we kind of, so we don't use so much ELISA, but we will basically go back and get glucagon for all of it. To answer your question about cell composition, this is what's so beautiful about humanislets.com is we didn't do the experiment, but it is done. So for all of these, there is a beta cell proportion done from a section from the block. There is estimation of cell proportions from the pseudo bulk stuff I don't understand. So for every donor, we have all of this information. And there's other groups around the world that get islets from Pat McDonald. He's now currently the largest supplier of research islets, certainly academic supplier on Earth. So we are convincing more and more groups to join with us, and maybe even groups who have their own sources of islets can all come on. And the more people do these experiments, put it together, it helps my research because I don't have time to stain and whatever. But it helps me answer your question when I come to Lille, which is great. Yes. Okay. It has been done.
What would be the time these animals on the
Yeah, I don't know. I mean, this was a study done by Robert Lustig, maybe 15, 20 years ago, specifically in a subset, I think, of adolescents with specifically some type of hypothalamic obesity. Like there was a very subset of people, and they did this experiment and it caused weight loss. It's one example. I think it's hard for big pharma, I think, to chase the idea of inhibiting insulin, especially like Novo that makes insulin. Maybe it doesn't look as good. Yeah. It's so confusing what's happening now as well. And now, of course, you have studies with glucagon promoting insulin secretion through GLP-1 receptors and vice versa and things like that coming out. So our classical idea of what regulates what in isolation maybe are starting to be changing. But I agree, somatostatin, and there's some nice papers that are starting to come out where people look at, I mean, obviously Patrick Horstman and Mark Housing and others are working on these to try to understand the role of somatostatin. To be honest, we haven't really gone there yet. We recognize that as a limitation and a weakness in our studies, but maybe someone will.
It's difficult because there's
no specific No, it's tough. Well, maybe this is what. One of the things that this project has also taught me is that we should start abandoning Western blots and ELISAs, and the mass spec is now so sensitive, and so we need very little sample, so cheap, either untargeted like what we did, or you can put in standards and you can do targeted mass spec. I think within a few years, we won't have ELISAs, we won't have Western blots. I've kind of banned Western blots from my lab anyway, because why do a Western blot one at a time when you can have 8,000 Western blots in 30 minutes? Oh, find out, I can tell you right now, the antibody's not specific. It's always not specific, right? No antibodies are really good, so. Yeah.
A lot to think about. So this change a little bit the paradigm from immunity into from putting insulin on top. But the first organ is the liver that feeds the insulin, right? And you show that it's also important your model with respect to steatosis. But I remember I don't know if they still talk about it, but in the old days, people like Chile and others were talking about the liver being selective insulin resistance. And that was a way to explain, for instance, the fact that dyslipidemia with insulin resistance obesity is sort of partly caused by the VLDL production by liver. And so I was just wondering if this concept of the so-called selective insulin resistance in the liver, and we didn't talk much about the dyslipidemia focused on glucose metabolism. I was just wondering what your thought is about that.
Yeah, I think it's a super important topic. So we've sort of mapped out the tissues that change at what rate. We think adipocytes change quite early. We think a lot of the effect in the liver is some of spillover, but at the same time, the liver has an enormous number of insulin receptors. And I don't believe that they're just there to suck insulin out of the portal system and degrade it. I think that's not physiology. So I think insulin, the liver is a very insulin sensitive organ. Obviously, it's there to shut down and glycogen release and whatnot. But I think this is a really important question. And I think we were talking earlier today, and there needs to be reanalysis, I think, and maybe using pig models or things like that of the insulin's action on the liver. There's some nice work that's been done in dogs. There's some nice stuff that's been done in mice. Of course, in mice, if you knock the insulin receptor out of the liver, it's one of the worst phenotypes. Interestingly, you knock it out of the muscle, almost nothing happens, right? And this contradicts our wide thinking that, of course, the muscle is an insulin-sensitive organ, but you have to think a little bit. You knock it out of the fat, it's kind of useful, but not too much. You actually knock it out of the beta cell and knock it out of the brain. So I think a lot about not just the tissues that are differently reactive, but the order in which the tissues, and maybe for different individuals and different subtypes of obesity and subtypes of diabetes and subtypes of metabolic liver failure, maybe the order is different. Maybe the paradigm is correct in some people and it's the opposite direction in other people. I don't know. The other thing that we talk about selective is the signaling pathways. And we're very interested in this. And I think this, I hinted at it in one of the questions. The dose at which insulin is active, at the very low doses that are physiologically relevant, it is a very powerful regulator of lipid homeostasis in small picomolar doses, in vitro and in vivo. And that's where it activates ERK signaling and MAP kinase signaling selectively in many models. If you have to go to quite high levels of insulin to get turning on AKT, in vitro, you actually have to go to quite high levels of insulin to get glucose transporters to move around. I think the signaling is different in low and high insulin, and the pathways are biased. And I think that's how some of these biased agonists may play a role as well. And that can help explain as well some of these paradoxes. It depends on how much insulin, and it depends on the duration, depends on whether it's oscillatory or still. It's so complicated, and we haven't even, we're very simplistic. We think, oh, insulin goes up, glucose goes down.
So in the knockout experiment, don't you think one potential flag is that most of these experiments are done in developmental knockout models? Nobody does it in the adult bioinducible And it's very difficult because you would use whatever cycling which improves up your microbiome. But I'm not sure there are very good reducible models.
No.
But
we're getting there. We're getting there. And we are now using virus a lot to do inducible models with locks. You can use CRISPR models. We did the inducible with the insulin gene, and it was interesting. It was a very selective adipocyte phenotype. And, you know, and glucose homeostasis was not touched. So I agree, actually, the development is really key. And that's a limitation of our studies as well.
So I may have the last question. If there is burning question, please go.
Yeah.
I have a question from the clinical perspective, so the whole animal, human, that's what we are looking at. You talk about differences as a whole, but of course there is fasting and there is postprandial, two very different phases. In your experiments, I would expect that basal and postprandial for post-stimulation couple together, but you will tell us. But in human, of course, postprandial, you have the gut. And we are strong believer that glucose absorption from the intestine is the main driver of the level of insulin very early after the meal. And we have many facts about that. But so my question is there any discrepancies that you looked at that between the fasting insulinism and the postprandial insulinism?
This is a great question, and I'm glad you asked, and I apologize for not really mentioning that. I think when we play with the insulin gene dosage, you know, they do go together, but mostly it's the fasting that is what we are studying in this. Now, I have collaborators like David Ludwig and others who are very interested in things like the glycemic index, and they're very interested in the insulin, they call it the carb insulin model hypothesis. And that's why they've engaged me in some of this stuff. They're very interested in, of course, why a high glycemic white bread or Coca-Cola or whatever is maybe driving obesity or insulin resistance more than rye bread or foie gras or something, escargot. By the way, I very much recommend. So, you know, I think it is very important now when we talk about hyperinsulinemia, actually, that we are just discussing, is it one? Is it basal? Is it postprandial? Is it both? And I guess to answer your question, our mice, mostly they travel together, but it's basal is the main thing we're studying. Then, of course, when we did this human islets and we stimulate, that is the postprandial response. I think it's very important to think about that in our diet as well. A healthy beta cell has a low insulin in between meals, a low fasting, and then it has the ability to make a response. The delta. In type 2 diabetes, the first phase comes down, but also the basal goes up. So the delta, the ability to register a signal is lower. And I think I also think, and I need to, of course, to spend more time thinking about this, because I apologize, I do tend to ignore the gut, but I agree with you 100% that a big piece of the puzzle of how fast something hits the beta cell, it has to go through, right? So I think there's a very big difference in a bite of food that is complex starches, carbohydrates that take an hour to dissolve and find their way in, and it comes to the beta cell gradually versus Coca-Cola or the profiterole I had last night that comes to the beta cell like wham, right? And it's the same amount of carbs, but how aggressive it is. And of course, there's the things in the order of the meal, whether there's already been protein before, after. These are very, very important. Yeah. Yeah, of course. Yeah, I mean, I didn't even mention this, but the beta cell's physiology, and a good friend and colleague, Christine Doucette in Manitoba, is working on this. The physiology of the beta cell flip-flop between the day and the night. There's a very important circadian rhythms. Yeah, it's been done in mice. Our human islet work really tricky. I think once they're in culture, they're kind of reset, but we don't know. We don't know if the donation, if the cause of death was that day or night, whether it sort of locked it into something. We don't know if they just had a big meal, they didn't have there's a lot of, some of the variability in the human islets, of course, comes from their prior physiology experience. Some comes from the separate those two. But it would be cool.
Thank you very much again, Dean.
Merci beaucoup.